Vanakkam makkale, let's get started. Today we are going to see about inflammation. Inflammation. Chapter number 3. Okay, in acute inflammation, there are three major components. Vasodilation, vascular changes, and leukocyte recruitment. Today we are going to discuss about leukocyte recruitment. It is a step-by-step process. First step is leukocyte adhesion to the endothelium. This is the endothelium and this is leukocyte. And treat this is where the leukocyte has broken. Treat this as the injury site. Okay. So these are the exact Robbins lines. The attachment of leukocyte to the endothelial cells is mediated by adhesion molecules whose expression is enhanced by cytokines. So as soon as an injury has happened, it will release cytokines, and in turn cytokines increase the expression of adhesion molecule. So it helps in attachment of leukocyte to the endothelial cells. There are two types of proteins for adhesion, two types of adhesion molecules. One is selectin and another one is integrin. Okay, this is first we will discuss about selectin. Just think about lock and key and consider selectin as lock. There are three types of selectin: L, P and E. P and E are expressed on endothelium, whereas L is expressed on leukocytes. Okay, P selectin, E selectin on endothelium, and L selectin is on leukocyte. And for this there is a common ligament called Cial-Lewis. I will write. For this there is a common ligand called Cial-Lewis. Okay, this is a ligand to which it get attached. So this is the selectin or key, and the ligands are selectin is the lock and the key is the ligand. Okay, it will keep the leukocyte to attach to. It helps in attaching the leukocyte to the endothelium molecule. It won't actually help in attachment, but it will reduce the reduce the speed of leukocyte. It will help in rolling of leukocyte. So injured cell release cytokines like TNF, tumor necrosis factor, then interleukin 1 and chemokines. These are chemoattractant cytokines. Okay, so within 1 to 2 hours it expresses selectin. And injured cell also release histamine and thrombin. Okay, so what to do? Histamine and thrombin will bring the visible bodies, that is a selectin or something like that, from granule to the cell surface. It means it is getting expressed here. The visible pallid bodies or selectins which gets to the cell surface from granules. So next we are going to discuss about integrins. Here also the key is the ligand is ICAM or VCAM. ICAM, it is a ligand for beta-2 integrin, LFA1 and MCA1. VCAM is a ligand for beta-1 integrin, VLA4. That's it. Cool. So this is LFA1, MCA1 for ICAM and VLA4 for VCAM. V for V. This is beta-1 integrin. Okay, don't forget this. It is a very important page. You have to remember everything from this page. Then the next second step is leukocyte migration through endothelium. Okay, it is also called as transmigration or diapedesis. So chemokines are attractant. They will attract the leukocyte towards it. So intercellular junction, this is where the endothelium has broken, right? This is the leukocyte, and these are called PCAM. These help in transmigration of leukocyte from the lumen to the injury site. Okay, it also produce these leukocyte produce collagenase, which pierces the basement membrane and enters the extravascular space. Okay, it produces collagenase, which pierces the basement membrane, and it enters the extravascular space, and it move according to the chemotactic gradient. So third step is chemotaxis of leukocytes. What it means, chemotaxis. Locomotion along a chemical gradient. It is known as chemotaxis. So there are chemoattractants. Two types of chemoattractant. It is like a magnet which attracts leukocyte towards it. Okay, now there are two type of chemoattractants, exogenous and endogenous. It's very important, just try to remember it. Okay, so in exogenous bacterial products, peptides like N-formyl methionine terminal amino acids, or exogenous. Okay, in endogenous, cytokines like interleukin 8, and in complementary system C5a, and in arachidonic acid LTB4. These three are very important, try to remember that. These are endogenous, which is produced inside the body, and exogenous, which are a foreign thing which act as chemoattractant. Bacterial products, peptides, and here interleukin 8, C5a, and LTB4. These are attached to G protein coupled receptor. So just we will once again, this is a picture taken from Robbins. First, this is leukocyte, and we already have silent Lewis X modified glycoprotein. This is a ligand for selectins. So normally it will go in the straight line, middle of the lumen. As soon as there is an injury happen, it releases cytokines like TNF and interleukin. It will help in expression of P-selectin, E-selectin, and L-selectin. And what happened? L-selectin actually is in low affinity only. After getting exposed to cytokines only, it will overexpressed, or high, it will enter high affinity state. Okay, first E-selectin, L-selectin. This is the ligand. The ligand is in leukocyte, so it will attract. See, this is E-selectin, and this is a ligand. They get attached, and it slows down. It slows down slowly, and this is ICAM. So this is where the integrin it's already present, but after cytokine, its affinity increase from low affinity status to high affinity status. So next step, it gets attached. This is ICAM, and these are ligand. It are getting attached. For ICAM, what is a ligand? LFA1 and MAC1, beta 2 integrins of these two. Okay, it will get attached, and it slows down slowly, and it will get attached. And these are PCAM1 or CD31, and transmigration happens here, and it will slowly move towards the chemotaxis, towards the injury site, and it will macrophage with microbes. It will macrophage the microbes, and that's it. This is inflammation, total inflammation. Then this is selectin. There are three types of selectin: L-selectin, E-selectin, and P-selectin. And these are the same for all the three. There is only one same ligament, common ligand. So not ligament, sorry, ligand. Then integrin. There is LFA1, MAC1. For these two, the ligand is ICAM1, and it is also CD54. Okay, ICAM2 is CD102. ICAM1 is CD54. ICAM2 is CD102. Then another one is VLA4 molecule, which for which the ligand is VCAM1. It is CD106. So 54, 102, 106. Try to remember this CD number. Okay. Then PCAM is there. It is CD31. Okay. That's it. Now it's question corner. Integrin ligands except. This is what just now I have said you to remember, right? Say CD, it's correct. It's correct. It's correct. This is the answer. This is the answer. Then what is CD51? CD51 is PCAM 1. It will help in transmigration. What are selectin ligands? What is ligand? Sorry. They are asking about ligands, selective ligands. Sial, Lewis, EX. This is the ligand. And selectin ligand L, P and E expressed on what? These two are expressed on endothelium. Then P is also expressed on platelet. And L is expressed on leukocyte. It's very easy, right? L for L, leukocyte. P for platelet. And E for endothelium. P is also expressed on endothelium, okay? Then chemoattractant, complement system. Which complement system factor acts as chemoattractant? C5A. And what is the cytokine? Interleukin 8. Okay. Then for Pseudomonas infection, what is the leukocyte which gets recruited for Pseudomonas infection? It is neutrophil. Okay. For viral infection, it is lymphocyte. For helminthic infection, it is helminthic infection and allergy. Just add this also, okay? Allergy. It is eosinophils. It's very important. Just try to remember this. And monocytes, after how many hours it will get activated? Monocytes will get activated after 24 to 48 hours of injury. And the neutrophils get activated within 6 to 24 hours. Okay. That's it. These are very, very high yielding questions. Just try to remember everything, okay? Just try to remember all these. Thank you.